But I'm excited to have Dr. Reid Nelson, who's a friend and colleague of mine when I was over at George Mason doing some aquatic research, which we'll talk about today. I'm just going to do the basic sort of introduction. Reid is an assistant professor in the Environmental Science and Policy Department at George Mason University. He's also a science fellow at the Potomac Education and Research Center down in Woodbridge. Yeah, you forgot you had that title, right?
Yeah.
Before coming to George Mason, he was a postdoc, a joint postdoc at NOAA and University of Santa Cruz. And then before that, he got his PhD at South Alabama and was an undergrad at UNC Wilmington. So he's generally from this area, grew up in Myrtle Beach, South Carolina, so an East Coaster. But with that, without further ado, I'll hand it over to Dr. Reid.
Excellent. Thank you, Travis. And thank you all for coming today to hear about my talk about the impacts of artificial light on predator-prey interactions and juvenile salmon survival. So this is work that I did during my postdoc in California, and hopefully I can bring some of this artificial light work here to the East Coast and to DC and the Potomac in general. So before we get into that part of the talk, I want to give you all a little bit of background about California salmon. So in California, we have 4 runs of Chinook salmon, and this is pretty much the furthest south that salmon goes. So they're already sort of at the edge of their range and their thermal tolerance for what they can handle. And so we have fall run, late fall run, and then winter run, spring run, and then the anadromous form of rainbow trout, which are steelhead. And so they differ both when they return to freshwater to spawn and then when they out migrate. And so I'll bring your attention to winter run here. Winter run's really interesting because they actually return to the furthest up the river system, and they return almost a full year before spawning, and then they hang out for a while and they spawn, and their offspring have to come all the way back down the river. Oh, whoops, I actually had bullets. And so they're actually broken into 3 evolutionary significant units with spring run, winter run, and then fall run and late fall run considered 1 ESU. And unfortunately, like anadromous species throughout many of our waterways, they are experiencing drastic population declines. Fall run and late fall run are species of concern. Winter run ESU is considered endangered. Spring run are threatened, and then steelhead are also threatened. So as you can see, populations really aren't doing that well. One of the big reasons for that is the lack of traditional spawning habitat, and this amount of spawning habitat is why these 4 runs were able to evolve. So basically, winter run would come in first, and they'd go all the way up into these tributaries you see as black now. Then you'd get spring run, and then you get fall run. And so the runs are actually named for when they come back to fresh water. But as you can see, the amount of freshwater habitat for these spawning populations has greatly decreased. So on this figure, everything in black is what's now inaccessible. And so this has basically shifted the population to be dominated by your fall run and late fall run, because they're the ones that enter the estuary last, and they're the ones that traditionally spawn in these valley rivers. Oops. So combined with the lack of spawning habitat, we're also in a warming trend throughout our climate, and California also is in pretty much drought every year. And a lot of people are saying that the drought out west is perhaps the new normal. I don't remember the exact numbers, but in the last decade, most of the years have been considered drought years. And so and there's also poor water quality in these systems. So unfortunately, all of these things combined, lack of spawning habitat, increased temperature, poor water quality, work against the native endangered salmon, and work for sort of your non-native predators. Your increased temperature stresses salmon to the point where they're at a higher risk of predation, and also increases the metabolism of your striped bass and your black bass species, largemouth bass and smallmouth bass, which are the voracious piscivores in the system. Just gonna hide this again. The other thing that's been going on is an increase in hatchery production. And basically this has resulted in increased fall run fish, and it's also weakened the portfolio effect in these salmon. So basically they don't have as much diversity across these different runs anymore, and they're more susceptible to impacts, especially if it impacts the fall run. A quick note on this: without hatchery production, California salmon probably wouldn't exist anymore anyways, especially for some really bad years. So there are some trade-offs there. There's a lot of research and some documentaries about this as well. But one of the things that contributes to their decline is actually poor outmigrant survival. So this is mortality in the freshwater stage of the juveniles as they're leaving the estuary. And this is because of increased predation, as I mentioned, poor water quality, high temperatures. And so predation is one of the things that people are really interested in. And that's because outmigrant survival can be really low, as low as less than 5%, depending on the year. And then predation by these non-native piscivores is a contributing factor. One of the things that my group was hired to study were different anthropogenic contact points. So basically places that aggregate both predators and prey that we've introduced into the river system. Some of the things we've studied were structure, just docks, bridges, those types of things, submerged aquatic vegetation, water diversions, and then finally artificial lighting at night. And so in terms of salmon risk and, well, fishes in general, lighting tends to aggregate fish on light. If anybody is an avid angler, you know that at night you can go target some of these lights and end up catching a lot of fish. There's actually these lights on the market to aggregate fish on your own docks. And then typically for piscivores, it increases predation success because they can actually see to feed. And then for juvenile salmon, it actually alters their migration because they've evolved to actually outmigrate at night when it's dark to avoid predation risk. And so they've shown that in systems that are illuminated, it'll actually cause the outmigrants to slow and pause. Okay, so all of these things, so we've established that salmon are at risk and they're at risk of predation. So this is one of the things we studied that might actually increase that predation. Beyond these types of impacts, direct impacts on salmon, artificial light in aquatic systems can alter spawning cues. It can alter the circadian rhythms of these fish. It can have quite a few impacts in aquatic systems. And you can think of it just like another pollutant, as you would like chemical pollution, but light pollution, just like we do in terrestrial systems a lot, where we think about this stressor. It's becoming more and more prevalent in aquatic systems. But for the purposes of this talk, I'm focusing pretty much on direct lethal impacts instead of sublethal like competition or oops. All right. So today we're going to talk about two different studies that we did to try to look at the impacts of artificial light in the California Central Valley, where salmon are both spawning and outmigrating. So we first wanted to see, did introducing light into the environment increase piscivorous fish density, both from an introduced source, which we did, and then an established source that's been in the system for a long time. And then we also wanted to see if light changed the relative predation risk of juvenile salmonids. So basically, does it increase your potential predators, and then does it increase the risk of your salmon? And so we did this at two different areas. We did this in the San Joaquin Delta, which is a tidal freshwater system. So basically tidal freshwater, it's completely fresh, but it does go up and down with the tides, experiencing highs and lows just like the coast. And then we did this at the Sundial Bridge, which is an iconic bridge in Northern California, situated on the fast-flowing Sacramento River. So two different systems, two different types of predators, but we have some really interesting results across both systems. So the first study I want to talk about is our study in the Sacramento San Joaquin Delta. And so this delta is a heavily modified system. On this figure, you're seeing what it looked like, sort of land use in the early 1800s and now in the early 2000s. Most of this delta has been developed for agriculture, right? So a lot of the water gets pumped out to use for irrigation, and then a lot of these land plots are actually used for ag. And it's all been leveed and channelized. So actually, when you're on the land, you're sitting below the water. It's pretty interesting out there. Kind of like a New Orleans situation, but there's a lot of water that's pumped out, and then also water from this system does get exported to other parts of the Central Valley, both for ag and drinking water. But it's also home to endangered and /threatened native fishes. I mentioned all our salmon. There's also a Delta smelt that lives here, and then as well non-native piscivores. And it's also the most invaded estuary in the world. So there's been a lot of changes, but it still supports productive aquatic life. So for this study, we focused on this small area of the Delta. We set up 5 different sites, and at these sites, we actually introduced our own light source. So most of these shorelines, the only illumination that was there would have been residual, just sort of ambient sky glow. Where we set up, we tried to pick sites that were relatively dark. We basically set up our own LED light. Throughout our study, we really only had predation at 2 of our sites, and this ended up being because of what was actually present in the water. But I'll get into that in a second. And then a lot of our study focused on this one site, L4, and that's because we ended up having a good predation response there, and potentially that was because it was a migratory route of striped bass, we think. Okay, so to get at predation, I want to go ahead and highlight what we were studying was relative predation risk, right? So we couldn't measure absolute predation because we were using tethering experiments. And so if you go back in the literature, tethering experiments have all these assumptions. So I want to be clear that it's relative predation. And while we are using a fixed individual that doesn't swim as well as something that's wild, it can help us study potential variables that may influence predation. And so we use these predation event recorders for this study. And this was sort of this is the second iteration of these predation event recorders, but functionally they're a large PVC pipe that suspends a smolt below the water surface. And as it goes down the water, it records GPS position and predation time. So this allows us to know where and when a predation event occurs on these tethered smolts that we can then correlate to environmental parameters like artificial light. The one thing I'll say about this technique that I think may provide some advantages over like fixed tethering experiments is that these are actually drifting in the water. So the fish is still swimming and moving throughout the river, potentially sampling a bunch of different habitats or environmental conditions for each deployment. And so this is just a quick video of a lot of GoPro work on these predation event recorders, just to demonstrate that they work pretty well. And we sample a whole suite of different predators with these. Mainly in these videos, it's striped bass and black bass species, but also some other syncharchids as well as catfish. Oop. Okay, just going to go on. What? Stop. Okay. So we use the predation event recorders to actually look at predation risk in the system. And then each night in our study site, we would use ARIS cameras to look at fish density. And so ARIS cameras are a sonar camera, but instead of using video, they use sound and produce a video-like image of the aquatic environment. And since we were working at night, we couldn't use traditional video. but you can see here a school of bait fish come by there, and you'll see them come back, and you see these two predators, two piscivorous fish chasing the school of bait fish. The only problem with this technique, or not, okay, so one of the problems with the ARIS cameras are that we don't know the actual species in the system. We can just sort of quantify, is it a large fish? And then depending the resolution that you're using, you can actually say, okay, we think it's this type of fish, but you can't usually get it to species. Sometimes you can get it to family. For our work, we were just simply looking at large fish above 200 millimeters, which we assumed were piscivorous. And so each night, this is what our experiment looked like. We'd go to a certain area of shoreline in the Delta. As I mentioned, we'd set up our light source, and we'd set up a 200-meter experimental run, basically, that we'd buoy off here. We'd have an ARIS camera in the light, an ARIS camera in the dark, and then we would run mPERS down this system throughout the night for about 4 hours. Then after the work was done, we'd actually come back through our experimental reach and quantify the light intensity, or lux. And we decided just to use lux because that's a measure of light intensity to the human eye. And for a lot of these species, they use the visible spectrum somewhat similar to what we do. So we thought that was a good measure of light intensity in the environment. So this is just some shots of us setting up our reach and then our light source. So you can see it's not a very, I mean, it's a bright light, but the actual extent of its influence isn't that large. Okay, so for our data analysis, for our ARIS, we use this fish density metric. Luckily, the guy we worked with for this work from the U.S. Bureau of Reclamation had like a nice workflow that basically took our ARIS data, filtered it, and quantified fish density all on our own. And so fish density was basically the number of fish pings greater than 200 millimeters within a 30-minute time band divided by area. But it's area on a 2D scale. It's 2D, not 3D, because we compress it on the computer. So it's a relative measure of what's there. The point is, it was a quick way to actually look at the amount of fish there without having to go through and count everything. So for this work, we wanted to see, each night, was there a difference between our light and dark treatments? Was there an effective time? And then was there any interaction between light as well as time? And then for our PER analysis, we used a Cox proportional hazards model. This is basically just an exponential regression, but it allows you to censor the data, used really commonly in medical studies. And so basically, when a fish gets hit, you can cut off the data there for each PER deployment. So what did we find? Well, the first thing we found is that if we look across all of our study sites combined, we do see a significantly higher amount of predators in our light treatments than in our dark treatments. However, we really don't see this start to separate until about halfway through our sampling nights, which was roughly around 2 hours, 2 to 3 hours past sunset there. And then, not surprisingly, this helped to explain why we didn't see a lot of predation or predation risk at a lot of our sites. There just wasn't fish there. There weren't predators there. Right? Regardless of whether there was light or not, there just weren't fish in the ecosystem to cue in on that light. And that was one of the reasons we ended up focusing on this site for a bit, because it wasn't so important to us. It wasn't so important to this how this worked across the whole ecosystem. We just wanted to see, did light cause an effect here? So pretty much, yeah, at about 3 hours through the night till the end of our study, light did increase predator density. And so as far as predation risk goes, you see a similar result. Early in the night, a lot of our predation event recorders didn't get hit, But then late in the night, as light intensity increases, the predation percentage also increases. This is set up like this. This is early night. This is late night here. Oh, wait, they can't see that on Zoom. So the left panel here is early night. The right panel is late in the night. Lux or light intensity is on the x-axis, and the y-axis is your predation risk. So basically anything above 1 is an increased risk. Anything below 1 is a decreased risk. And although the confidence level is really wide on this exponential response, you can see that for our late in the night data, as light intensity increases, risk also increases. And then perhaps our strongest predictor was, regardless of whether it was early or late in the night, if predators were present, the risk of predation increased, not surprisingly. But this highlights the two things we think were going on here. And so this is just a extrapolation where we basically smoothed all of our light values together. And if you see here, Lux, this is our light intensity for study reach. This is the proportion of PERS that were predated upon, both late in the night and then all night. And so you can see, even with this visual metric, it seems to be centered around our light source. So basically with this study, we found two things happening. So when we increased light intensity in the environment, it increased predator density, and increasing predator density also increased predation risk of the salmon. So that's basically what we think is going on here, is when there's light in the environment, predators are attracted to it. And because of that increased light, they can also forage more effectively. So we didn't actually separate those two things, but we think it's a combination of increased predator density and increased foraging efficiency. However, with this study, we only saw an effect, and significant effect, pretty much 3 hours after sunset and later. And in terms of management, this could be really important, because a lot of times early in the night, is when we need the artificial light more in our cities. We need it for safety. We need it for recreation purposes. As the night goes on, more and more people are going to bed, and they don't need it as much. So we made the recommendation that perhaps we could come up with sort of late night lighting reduction schemes to potentially increase survival. Okay, so quickly, we're going to talk about the next study we did, and this was at an iconic pedestrian bridge in Redding, California. It's called the Sundial Bridge, and that's because when the sun hits it, it actually creates a very large sundial on the land here. And this bridge, it was made in 2004, and as soon as it was made, they started getting dinged for having too much light on the river, and they said that it was causing salmon mortality. And they said this was due to rainbow trout predators. And this actually led to a revamping of the lighting in 2014 without any studies, really. People went out, they measured the light, they said, there's a lot of light here, we should change the light. So they went from halogen lights to LEDs, and they said this will help increase salmon survival. They also entered agreement with the California Department of Fish and Wildlife to use a minimum lighting scheme from August 1st to February 1st each year during the outmigration of winter run. And the reason this bridge created such a controversy is that winter run, the endangered ESU, can only spawn basically from the Keswick Dam. They only have about 10 kilometers of spawning habitat left on the Sacramento River. And the Sundial Bridge is smack dab in the middle of this spawning habitat. So traditionally, right, they could go above Redding into all these streams and tributaries. Now they can't. They're stuck here. And so that's one of the reasons that this created such a stir, and one of the reasons that we were brought in to look at this. So the city of Redding, California was nice enough to let us take full control of their bridge. They gave us a laptop, and it basically had, I don't know if anybody ever does any DJing, but it basically had like a light program that you would have, like a DJ would have. And so you could change all these channels. There were a couple nights we got a little crazy, but we're not going to tell anybody about, like, after our experiments were concluded. But to be simple, we basically set 4 different lighting treatments on their bridge: a no light treatment, a 25% light treatment, 50% light treatment, and 100% light treatment. And we did this for white light of all their channels. I wanted to take the time to test different spectrums of light, but we just didn't have the time to do that. For this work, we made a new iteration of our predation event recorders. And if anybody's interested, I've got one you all can look at. But basically with these, we didn't want to release things into the environment and then have to go get it, because the river moves so fast that we were like, we'll never catch these. So I came up with these. They're based on Arduino coding, if anybody is familiar with that. And all they do is they record GPS position every second, and they record whether a predation event has been hit or not. So basically the salmon's here. When it gets eaten, it pulls this magnet, which trips a reed switch. And there's an SD card in there logging everything for you. And just so you all can see how it works. So this is a shot. This is from a different project, but this is a shot of one of these on the ARIS here. If you can see where the pointer is, this is the micro-PER. This is the prey fish swimming around here. And then here comes a bass, most likely. It took this fish a while to get the smolt off of this predation event recorder, but this showed us that this worked well, and it was a valid technique. Unfortunately, this wasn't in this system in California. And so for this work, we did a similar gamma model, and I'll mention here that the reason I decided to use this is because if you fit this in a normal, like, linear model, all your predictions that came out were negative, and you can't have negative fish density. So it made a lot more sense to go this route. We tested both within river banks and among river banks. So we wanted to know, we set up ARISes on both river right and river left. So we were curious, does light have an effect? And is there a difference among river banks? And then we had to include quite a few random effects, because we had continuous data recorded throughout the night for like 7 hours at a time. So it wasn't independent. So our actual replication was only like, I think we did it over 5 weeks. So we only had 5 replicates for each light treatment here. And then we also wanted to look at how fish in the system responded to light. So we actually use the segmented function, which actually looks for significant breakpoints in the data and then fits models to it. And then we also, for our last week of sampling, did continuous ARIS footage throughout the entire week. So we wanted to see, is there any diurnal patterns to fish density at these bridges? For a micro-PER analysis, I was really proud of what we did. We only had 4 events, so there was really no predation. As I mentioned, this worked really great somewhere else. It didn't work in this system. And I think it's because the predators, the rainbow trout we were looking at, just really aren't piscivorous in this area. So here's the results of the micro-PER. They worked really well. GPS positions looked great. These are all of our sampling. Like I said, unfortunately, we just didn't have a lot of predation. Part of the reason for that is I truly think the rainbow trout aren't piscivorous. So I set up tethered fish fixed in the bottom just to see, can I get these fish to eat a salmon smolt? Multiple days of this, no fish would eat a salmon smolt. So what do we find? Well, when we actually look at our ARIS data, across our light treatments, we do find that on river right, Any light into the system across all our treatments is significantly greater fish density than no light. Interestingly, we didn't see any significant differences on river left. And then whenever there was light in the system, river right also had higher fish density than river left. So when light was in the system, it increased rainbow trout density. And we're assuming these were rainbow trout here just based on our own visual observations and talking to state biologists about what could be there. But when light was in the system, rainbow trout density increased on river right, and there really wasn't an effect on river left. But even with this increased potential predator density, like I said, we didn't have increased predation on our sampling metric. Just to give you all an idea of how many fish we would see each night. So this is a shot from river right. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, I don't know, 16 or 17 fish just from a random frame. And these fish would queue in on this light and hold there. It's like this all night. It was pretty impressive on that side of the river. And this is what the diurnal patterns look like too. So similar results to just our night analysis. But if we look at this through time, the x-axis here is minutes past sunset. The y-axis is rainbow trout density. You can see that, well, two things become really interesting here. It looks like fish are queuing into riverbanks right around sunset. So it looks like fish are showing up on our ARIS cameras. Our ARIS cameras were set pretty close to the riverbank. So it seems that fish are potentially moving to shallower water right around sunset. This density rapidly increases till about an hour past sunset, till it reaches pretty much an equilibrium for the remainder of the night. And you can see, although this pattern occurs even when no light is present on river right, the response seems to be greater when light is present. And then on river left, even though there's no difference of light, what we're seeing happening is still sort of a queuing in, fish density increasing during this time. This is what our continuous data looks like. So sort of the same trends. This is on river right. Basically the gray areas, the period of sunset to sunrise. As soon as the sun set, pretty much great increase in fish density, and then it would drop out at sunrise each day. The other thing I'd like to mention on this figure here is that this day was a 0% light treatment. So you can see this is our experimental period where we didn't have the bridge lights on. And then at the end of the night, once we were done experimenting, the city wanted us to turn the lights back on. So once we turn the lights on in this section here, this is the new response later in the night. And so while we weren't testing that, this even demonstrates that within night, turning that bridge light on seems to have an effect on fish density. So what do we think is going on here? As I mentioned, I do think that trout are moving to the river margins during twilight. You know, as that light's decreasing, a few things are happening. Invertebrate drift is increasing. It's a really popular fishing time. You know, a lot of people start coming out and fishing your margins here during that time. So I think trout are already moving to the river margins during twilight, regardless of whether there's light or not. But I think when the light is present, it further increases foraging success of moving to these margin habitats. And the reason I think we saw more fish on river right, which in this figure is the bottom, is because it was a little bit shallower and a little bit slower flowing water. So it wasn't as stressful to maintain position and a little bit easier to acquire prey on this side of the river. Now, if you remember, we didn't see an increase of predation even on this side. And that's because I think truly in this system of river, there's a lot of research that shows these trout just aren't really very piscivorous. Wild trout can be piscivorous. In this system where invertebrate density is really high, there's a large invertebrate drift. We think that they're loading up on invertebrates, and they're not they're just not eating fish. And that's why I don't think we saw a response with our data here. And we do think that based on this, the river right was probably giving more favorable foraging habitat. Right, so in this study, increased light seemed to increase predator density. And there was an impact of river habitat, most likely. And we think this is likely, it probably increased invertebrate predation, but it didn't increase salmon predation. And so we didn't actually study this, but we think this is what's going on in this system. The other interesting thing to think about with these rainbow trout are sublethal impacts, right? So this is a really popular rainbow trout fishery. It's a trophy fishery. You know, I think average trout are greater than 18 inches long, which pretty big trout. And so it could be that if the light's attracting too many fish, then prey is available. There could be adverse competitive interactions. There could be potential other sublethal impacts on the trout, too, that we didn't study here. But what we did show is a pretty clear response of fish to light in a completely different habitat. So just to recap, in our Delta study, increased light did increase predation risk as well as predator density, but that only occurred late in the night. As I mentioned, I think a good solution to that could be to decrease Allen late in the night, you know, when people aren't using as much light. Then in our Sundial study, we did see increasing light, increased fish density, but no predation effects the way we were studying that. We could have been biased the way we were studying it. There were out-migrating salmon in the system at the time as well that I noticed actually on river margins, not actually in the river channel. So we could have had some issues there as well. But potentially, at least as far as the city is concerned, they don't think that their bridge is having population level impacts. And we didn't really say that. We just presented, here's what we found. We'll let you all make your own decisions. But I do think there could be some sublethal impacts on a valuable recreational fishery for rainbow trout. One of the reasons we think this is the case, as I mentioned, is that in the Delta, we have a lot more piscivorous fishes because there's striped bass and largemouth bass, while upriver, we really have these invertebrate predators. And this isn't to say that later on, you know, in a different season, a different salmon run, striped bass do come up that far. There could be a great impact of that bridge, but at the time we studied, we just didn't see it. Some of the things we're doing in the future there is we are actually trying to now say, okay, we know in the Sacramento San Joaquin Delta, increasing light, increased predation risk. If we take this and convert it to a survival estimate based on the amount of illumination on the river throughout the whole system, what is the actual mortality if we were to decrease intensity by half, if we were to take everything out completely? So we're trying to get at what would the population level impact be? So what sort of benefit could we get if we remove lights and it does increase survival? What benefit can we get? We're also, like I mentioned, looking into other contact points in the system because salmon survival is so poor, and we're pretty much trying to find any way to increase out migrant survival at this point. Another thing that I thought was crazy out there is that everybody cites that predation mortality is high. However, there isn't really a good actual estimate quantifying that, right? Because predation's hard to observe, it's hard to quantify. So we were just funded to actually look at predation mortality in the Delta using a new predation / temperature acoustic tag. So basically we have a study supposed to happen this spring. It's probably not going to happen until next spring, where we're tagging a lot of steelhead smolts with these tags, releasing them in a stretch of river that we have a lot of acoustic receivers. And then based on our tracking methods and what the tags tell us, we should be able to get a mortality estimate of fish that were eaten by piscivores fish that died of natural mortality, fish that lived, and then potentially fish removed from the system by avian or other terrestrial predators. If this works, should work. I promise the state of California it would work. If this works, I'm hoping to take this technique to something like our light setup and release, tagged fish in illuminated treatments and unilluminated treatments to actually try to get at, instead of a relative predation risk, actually get more of a quantitative estimate of predation. Briefly before I stop, I just want to talk about some of the work we have going on here in the area. I would like to bring some of our artificial light work here to DC. I think there could be some potential looking at river herring predation risk in our spawning systems here. This is an imperiled and anadromous fish here on the East Coast. I think this could be a really interesting future research direction. Currently, we're actually looking at river herring migration diversity in the Potomac, and this is super fresh, but based on the chemical profiles of their otoliths, it looks like blueback herring aren't really as anadromous as we might have thought. We might have more resident population. Like I said, this is super new. I'm trying to look at contaminants of emerging concern in fishes. We actually submitted a proposal yesterday looking at fish downstream of wastewater treatment plants to look at impacts of these contaminants. I do some long-term ecosystem monitoring in tidal tributaries downstream of the wastewater treatment for Alexandria and the county of Fairfax. Working trying to look at striped bass post-release mortality here in the Chesapeake Bay, working with some folks on eDNA profiles of the Potomac River. And then I've got a student actually looking at the effectiveness of ITQs, or individual transferable quotas, in fisheries management. So I'm not going into a lot of detail here, but if anyone has questions about this or wants to collaborate, please let me know. I'm looking for trying to get my foot in the door here back on the East Coast. Yeah, so with that, I'll take any questions.
So I have a question, or maybe a set of questions.
Yeah, perfect.
First thing, it reminded me of Shark Week a little bit, because I think I saw something about South Africa and the lights that's produced there and how it changes great white feeding and stuff. It's kind of a similar concept. But what I was thinking is that in it seemed like maybe the connection in both so the later data sets you showed from the Delta and the Sundial studies, that it was the invertebrates that that's what I was trying to understand, is that the invertebrates that are showing up in both cases that are for the Delta, the invertebrates are attracted to the lights, and then the smaller fish are attracted to them, which means the predators are attracted to the fish. And then in the other case with the Sundial study, the rainbow trout are, like you said, are probably attracted to the invertebrates that are there. So it's like the invertebrates that are attracted to light. That's sort of, is that your take on it? Or
It could be, right? So it could be like a bottom-up process, right? Where the bottom of the, even, well, I will say even zooplankton, but actually light messes with zooplankton. They actually won't come to the surface when artificial light's present. So we don't actually know. And I think, yeah. So is it that smaller fish are attracted to the light and bringing in bigger fish because of the smaller fish, or bigger fish attracted to the light because of increased predation success? I don't actually know, but both of those mechanisms could be happening.
And, so, and it made me think, like, with your with the Arduino setup and like that type of method, you can look like it was mostly set up for sampling like fish. But I was wondering if you could set something up to where there was invertebrates as the bait. And I don't know how that, like, I could almost imagine like a little packet of invertebrates that you have.
So,
could be, I know that that's not, ideal.
So we tried, I didn't talk about this because it didn't work either.
Yeah.
So we tried a couple nights doing artificial flies.
Okay.
Yeah. Like invertebrate. We cut the hooks off.
You mean like with the rod?
Yeah, like caddis. caddis and midges and different things, and we suspended those from these
Yeah.
to see if we'd get a response,
Mhm.
but we just weren't. And part of it could be that we were sampling at depth and not on the surface.
Yeah.
And a lot of those fish, and when those invertebrates drift, they release, that come to the surface. And so you were seeing a lot of fish queuing in on the surface. So that could have been a sampling problem.
Yeah.
But yeah, it would be really interesting to see, right? We were very fish focused because that was also what the funder wanted.
Yeah, no, it's a cool question. And then, the other thing is, the otoliths. Did you look at the otolith composition at all in your California studies? Or is that just
So-
we'll get into now here?
Yeah, well, so there are, yeah, so I'm a big otolith guy. In fact, more of my research has been done on otoliths than probably anything.
Yeah, 'cause I'm curious about if you look at heavy metals in the otoliths or anything like
So a lot of that, okay, so some people have successfully detected heavy metals in otoliths. The problem is that a lot of those heavy metals are bound elsewhere in the fish,
that.
right? They'll get bound in the liver, they'll get bound in the tissue. They won't actually stay in the blood and then make it through the barrier into the endolith and then actually get deposited in the otolith. So when they are deposited, it's in really trace amounts, and it's kind of like lead is one that's been
Yeah.
able to be detected some
Mhm.
by some studies. What I'm mainly using it for is to look at migration diversity.
Okay,
So over salinity gradients, right? So strontium
right.
is
yeah.
really similar to calcium. It substitutes really well. Barium also substitutes really well. So you can actually use those two to look at habitat movement. Like I said, there's been a little bit of success with lead, but yeah.
So I had a question. Fish are kind of clever and they learn. Is it possible that the same predator ate all of your fish and it took them 3 hours to figure it out? So the early lights, they were just kind of figuring things, and then they realized if they queued up there, they do very well. Wait for this thing to go through them.
So you're exactly right. It could have been, now it was definitely more than one,
Right.
but it could have been the same school of fish queuing in on the light. We thought that we would have seen that. I guess if it was a learned behavior, it was such a short time, but we kind of thought that if we saw that, we would have seen it lose its effect as the experiment went on. Right? Potentially they would queue in, they would learn each night, but maybe it just took 3 hours each night to queue in, and then, maybe there was a within night learned behavior, not a across night. And we actually, with the Sundial project, that's one of the reasons I thought that fish might still be queuing in on the bridge even when lights aren't present. But it could also be that they're just queuing in on river margins. We didn't actually test it. We could have moved it. What we should have done, if we had more time, we would have moved the cameras further downstream and see if we saw a similar response in a different area.
Well, when you put your radio tags in, it would be interesting to see multiple fish.
Yeah, like how many we get loaded up. Yeah. Yeah, so that's something we couldn't separate, right? We don't have individual two with the ARIS. We don't know. Yeah.
I'm curious about the temporal scale of the lights. Like obviously the bridge has light on all the time. But if it was a vehicular bridge and it didn't have lights on it, would a couple hours of nighttime bright headlights driving over, would that still have the same effect? Or would it, is that just too brief of a time to really have that effect?
So that's a really good question. My guess would be if it's only a few vehicles, probably wouldn't be in the system long enough to create any impact. But if it was pretty consistent, right? So you always had quite a bit of light intensity coming off of it. The other thing you see on bridges is also just illumination for water, for navigation purposes. So they have shown, I think it was in somewhere in the Pacific Northwest, I don't remember the river, but they showed, I believe it was seals, skin, not a mammal guy, but they showed, I think it was harbor seals, maybe queuing in on a bridge lighting, eating salmon. And then when they turned off the lights, the seals, the predation pressure went down a little bit, but then they sort of queued back in eventually anyways. But there has been some higher organism effects, but in terms of how much light it takes, It's a really good question. You know, in this study, it also didn't take much light to, you know, even at our lowest light treatment, right? Which is kind of what they set their management protocol to. We still saw a response. If you look at some of the laboratory studies, even really, really low amounts of light will increase feeding success. But that's, you know, I'm not really positive how long it would take for this sort of attraction to set up. Be a fun experiment. You could put a light in and look at how long it takes for fish to sort of cue in on it.
Thank you.
Yeah, you're welcome.
I have more of a policy question. So on the Sundial Bridge, you didn't see predation on the fish that they cared about, but you saw a change in the ecology of the system. Did they at all care about that? It was just like no predation, so we're good.
Yeah. And truthfully, I left before any of this. Right, that paper was published last week, so I don't even know. Like I don't really know how it's being used right now. I sort of jumped like out of that world, but yeah, no one. So the city was pretty like upset, right? So I spent a lot of time meeting with the city because they'd already spent, I don't know, probably $1 million or $2 million, like revamping the lights, changing it all, doing all this complicated lighting management, and yeah, and it just. They're upset because, I mean, it's a cool bridge, like just the aesthetics of it. And they can put lights for like breast cancer awareness. Like when we were there, it was lit up blue for COVID. So it's a really big draw for the city. And so if they can say, oh, well, we're not hurting this endangered salmon, cool, that's all that matters. And it's such a trophy fishery there anyways. I don't think they're that worried about the actual ecology of the system.
Are the rainbow trout, hatchery fish?
Not there. Not there. Yeah. Good question. Though. One of the reviewers actually asked the same thing.
So you have, I don't remember what it's called, but
Yeah.
yes, that. So that one was not, you said because you didn't want to go and retreat it, was that somehow tied?
Yeah. So basically, we just tied this to a fishing pole, and because the river was moving so fast, we casted it upstream of the influence of the bridge lights, and then just let it free spool downstream.
Where do you use that technique?
And then we bring it back, and so we actually recorded start and stop time on our cell phone. And then, because what's happening is basically once this thing turns on and grabs GPS, it just records its position every second, and then it records whether this has been pulled or not. And so then we had a start and stop time, which we filtered out. We were able to take that whole massive data, run it through a script, and just say, okay, look for start and stop, pull it out, did a predation event occur? So those drifts were anywhere from like 1 to 3 minutes. They were fast. I mean, that river is like cruising.
So I guess like, I'm not.
No. Keep going. This is great. Absolutely. Right. Like it could have been. So when this worked really well, it was in a slow flowing tidal freshwater system. Right. So there is some potential sampling bias in that system. We don't have a good handle on what was going on underneath either. You know, we probably had like, there was a couple meters of leader on this thing. And so, yeah, there was a lot of uncertainty to that part. We thought that we would, even with all that, we thought we would at least see a little bit, right? More than 4 over like 1,400 deployments, if there was a thing.
You mentioned that those floated down for like a minute, but in the video you showed how the predator fish was having trouble getting the magnet out. So how long did it take that fish to get the magnet out? Is it possible they just didn't have enough time before you put it back in? Or was it like only 30 seconds?
So when we pulled it back in, we let them, so since we were only studying, it was way past what we are actually interested in before we ever brought it back. But it could be, but we didn't even notice, most of what we got back didn't even look like it had been touched. Right. So sometimes, that's a good point. Sometimes you'll get it back and your tethered prey will be dead, or it'll be missing half its body, or like, and even if that happens, usually it trips. So usually the first hit will trip the magnet. And then the predator will be frustrated because the fish is tethered through its operculum with fishing line. So it's sitting there pulling on it, trying to get, so a lot of times you'll get them back with like a dead smolt hanging off that actually has bite marks on it. So you can tell that it's been hit. But it's a fair point. Like it was one of the things we struggled with the whole time. Like this river's moving really quick. Is this a long enough time to sample what we want? You know, it's one of those trade-offs we decided, we could have tried to fix fish, but then when they got in the current, they were just like they were just being pulled. So
I've heard about invasive fish species around here. We've got snakehead, but I guess they're catfish. And could you use lights to like draw those over and then harvest them? I'm not a fish guy, but imagining.
yeah, potentially. So there are.
Controlling invasive fish somehow.
So there are fisheries that use light to attract. I mean, a squid. I mean, it's not a fish, but a squid fishery. They use light, and the squid just jump onto it. Bait fish are attracted to light. So there might be some applicability for that. Right now, it seems like there's open season electrofishing fisheries. So that might be more. And I haven't ever thought about it this way, but a lot of the guys that harvest snakehead do so on boats at night with really bright light to illuminate the water. So while they're using it for their own purposes to see the fish, I wonder if there's actually a bit of an attractive effect as well.
That might be an angle you can talk about controlling invasive species. And, assistant professor looking for things to get used to. Get in with some local agencies.
Yeah. Yeah, that's a good point.
Well, let's give Dr. Nelson another.
Thank you.